Thank you very much. Thanks everybody for coming. Thanks to LGC for inviting me to provide this talk. They've been absolutely fantastic at supporting me in essentially using some of these technologies that are used in a lot of the other crops, as we see in cotton, and now transferring that to the cannabis space, which is a relatively new, given the fact that it was recently legalized in Canada, where I'm from, in October 17th last year. So lots of effort in trying to catch up on a lot of the very creative breeding efforts that have been going on. And before I continue, I'd like to also highlight the fact that this particular study would not have been possible without the efforts of my colleagues from Las Vegas, Digipath Labs, that have provided a lot of the phenotyping information, the chemotyping information in particular, for Cannabis Accessions in the medical market in Nevada. So before I go too deep into what we've done, I just want to give a little bit of background about cannabis as a species. If you look in the literature, you'll find that there's varying descriptions of how cannabis is described botanically in terms of having two different species. I'm a biodiversity researcher. I view this in a much more dynamic fashion than most species concept, and I do view cannabis as a single species. And for the sake of this talk, I will refer to mostly the gene pools of cannabis that essentially are much influenced by humanity as well. So if you look at this map, you've got the overall morphological description of what the species or the gene pools look like. So you've got the sativa, very tall, a tall plant. You've got an indica, much shorter, much denser inflorescences, and then you've got ruderalis, which is thought of as a spontaneous variety, very short. I've witnessed some of those in Eastern Europe growing spontaneously, and they're very short and very underdeveloped. But essentially, we have four main gene pools where we're talking about European hemp, northern leaf hemp. It's summarized there with NLH. And then the drug type cannabis, where we have broadleaf and narrowleaf drug types, which are often referred to as indica sativa, and when we're talking about the commercial varieties that we talk about. But essentially, these are mainly hybrid of each other anyways, which I'll get to in a minute. And something we don't talk about so much is the broadleaf hemp, so the Chinese hemp, which essentially is a non-psychoactive fiber type varieties with broadleaves, which I unfortunately have never had the chance to run a sample of. But as I was saying, these species have moved about. These species have moved about the globe, most likely with humanity, and probably also in ancient times, where currently what we find in Europe or in European hemp are possibly hybrids of the two original gene pools of broadleaf and narrowleaf hemps. And then what we refer to, or at some point was referred to as marijuana, most likely a hybrid of the drug type varieties. And more recently, what we've seen is a strong push for less psychoactive varieties that express CBD as opposed to THC, and those are most likely caused by introgression of the CBD acid synthase gene from European hemp into a drug type cultivar, kind of as we saw with the talk from Robert, where we're trying to introgress something from a wild species into a more commercial type species. So what this paper shows here is essentially the divergent selection that has occurred, possibly from a common ancestor, leading to fiber type cultivars and drug type cultivars. One of them focusing on either hemp nut production or fiber production, and on the other hand, resin production, particularly inflorescence, large inflorescence with a large density of glandular trichomes that produce the secondary metabolites that have recreational and medicinal properties. Essentially, to show the difference between these two gene pools, we have here an example of the comparison of density and also trichome size between fiber type and drug type varieties. And you can see here that you have about 160 or 1.6 times larger trichomes found on average in drug type cultivars than you find in in fiber type varieties. And this is, most likely due to the divergent selection for these particular traits, where the genes underlying the production of cannabinoids and resin production haven't been favored or haven't been selected for it in fiber type varieties. So the title of my talk is about chemovars, and chemovars really are means to classify varieties based on the chemical expression. What's quite interesting, cannabis expresses cannabinoids as well as terpenoids. Cannabinoids have no scent or smells, so very often when we think of cannabis, we have a particular smell in mind, which is most likely caused by the terpene expression of the plants. So what this graph shows here is work done with DigiPath, which right now the data set has about 9,000 entries that are chemotyped at 11 cannabinoids and 19 terpenoids. But what's kind of interesting is that because a very strong focus has been placed on cannabinoid expression, it's not a valid means to classify varieties based on where they came from or where they originated, because essentially that was the one trait that it's selected for, and so there's very, very strong breeding efforts for introgressing particular cannabinoids into different lines. So we chose to get rid of cannabinoids, although they're shown here on this map. But what you can see, this is a principal component analysis of about 5,000 data points. And we were interested in seeing how they clustered based on the chemotypic information. So what we found is that there are three major groups, and this is just drug-type cannabis, so high resin producing medicinal commercial varieties. These varieties cluster into three major groups. The first group being dominated by beta-myrcene, which is also one of the most, so myrcene is the most common terpene found in cannabis, and it also is the main constituent of the essential oils of fiber hemp. So quite an ancestral, or I believe it's an ancestral terpene. Another group which is dominated by terpinolene. And then a third group where the dominant terpene is limonene and beta-caryophyllene. And essentially, all the arrows are the variables that show how these different groups cluster, and the different colors here correspond to each of those three groups. So what we did, and here is the part where LGC comes in terms of moving from very large panels generated by genotyping by sequencing. This was actually outsourced to a group from Boston, Massachusetts, called Medicinal Genomics Corporation. They offered that as a service where they would sample a plant and they would return a large SNP data set. I think this, essentially what we did here is take 70 accessions that were genotyped at about 180,000 single nucleotide polymorphisms. When those were filtered, we came out with a panel of about 1,500 SNPs, or in this case, 1,409 high-quality SNPs. And the idea was to now constrain the analysis. So in this case here, again, a principal component analysis that's constrained based on the chemotype profiles that I showed you earlier. So based on the different expression of beta-myrcene, limonene, and terpinolene. So you see here essentially that translating quite well based on these 1,400 SNPs. And essentially what you can do with these analyses is you can extract the loadings that will give you information on which particular SNP is driving the structure that you're observing here. So it's a little bit of a statistical trick that we used, but essentially we decided that we were going to focus on less than 50 markers. So we selected 49 of these 1,400 SNPs. So 48 of them that were the most informative based on the statistics, and we sent them to LGC in Massachusetts as well, where they essentially screened our population and additional individuals at these 48 CAS assays. And Robert did a really good job at describing how these work. After sending those out, one thing I should tell you guys is that this was based on the cannabis genome that was published in 2011. So in November last year, we had 2 new versions of the cannabis genome that were published, much more extensive, where the linkage groups are segregated into 10 chromosomes with a much higher coverage. So in this case here, linkage groups were something like 135,000 contigs, so much and very incomplete. So we don't have a full picture of this, but in any case, we decided to design CASP assays and test them and see how they work out. Turned out that 9 out of the 48 failed to amplify or provide any amplification with this technique. Reason for that being a very scattered genome that we used, which was about 6 years old or 7 years old at the time, and has been recently replaced. We found that there was some discrepancies between the genotype by sequencing approach. So in 16 cases, 16 SNPs that were shown to be polymorphic based on genotyping by sequencing approaches were actually monomorphic, even when we looked at European hemp versus medicinal cannabis. So that speaks again to the quality of the initial assembly that we use, most likely, where essentially I would say the in silico data was most likely wrong, and I trust the wet lab part more. But all in all, we came out with 23 polymorphic markers, so a little less than 50% of these markers came out as validated. 18 of them were associated with this terpene model, and an additional 3 were, I would say, kind of custom markers that we added in because we were interested, obviously, in THC expression. So 3 SNPs associated with the THC acid synthase, 1 SNP in the albumin, essentially for the seed production, and then 1 SNP associated with the mitochondria, which we use more for essentially teasing apart these large groups between each other. Here's just an example of a comparison. So just to show the performance of these SNPs and how they work when you're trying to assign these accessions back to their Chemovar cluster. So you see that essentially in this case is I think 1 individual that's cannabis assigned, 50% to another group, but 69 of the other individuals actually with genotyping at 18 SNPs were actually assigned to the correct group. So this is something that's kind of interesting. It's also been mirrored in other jurisdictions. So here's a study that was done in New Mexico, where they do show kind of that similar myrcene, terpinolene, and limonene clustering. And here's an example of a data set that is still work in progress, but essentially where we took this panel and we genotyped a large pool of germplasm from a Canadian licensed producer called the Flower Corporation in Kelowna, British Columbia, where we're from. And some of the interesting things to see here, you can see in pink on the top left, European hemp, that clusters together. There's a little pink line there. And then essentially, even in a totally independent data set, so this is British Columbia as opposed to Nevada, where the initial SNP markers were developed, we still see that same clustering when you go back in terms of these 3 major groups, myrcene, limonene, and terpinolene expressing cannabis. You see a little green cloud there, and that refers to a variety that clusters with the myrcene dominant varieties, but actually has alpha pinene as its main terpene. And again, this was something that was confirmed in New Mexico, where pinene and myrcene are always found associated with each other. Something which is quite interesting with these 18 markers is obviously, it's a very small number of marker. It's a cheap technology that we can implement in-house, and very often even in the field, depending on the machines that we have available. And it offers essentially an individual barcode for every variety that we have, that anybody grows. So it offers, it enables us to now, have some transparency in terms of the origin of different variants. The same as when we're talking about now products going from a producer to a retailer we have the ability to track these varieties in a very simple system, as opposed to using more advanced sequencing approaches. Essentially, here's another example of how these markers are clustering based on the position of each individual. And so you're able to actually find different markers that are associated with, or a particular allele associated, for example, with hemp. So if you look on the top left there, we've got a THC allele, a THC synthase allele that's always homozygous in hemp and is essentially an old drug-type cannabis homozygous for the other allele. As well as a mitochondrial marker. So that enables the identification of not only whether or not the plant is going to express CBD or THC, so whether it's a psychoactive or non-psychoactive variety, but also very targeted tests. We're using only 2 SNPs to tease apart CBD expressing fiber types versus CBD expressing drug types. And this can be assessed in vegetative varieties. So you don't have to wait the full 12 weeks it takes to grow a plant and then flower it to have essentially the expression of cannabinoids in the flower. You can actually do that in the seedling stage. So when we're talking about accelerated breeding and some of these things that Robert brought up here, where we're trying to integrate a particular part of the genome into a commercial variety, for example, you're able to do this with this type of approach. So recently it was published that both CBD and THC synthase genes are found on chromosome 9, but that there are other loci found on other chromosomes that affect the overall quantity of cannabinoids being expressed. So with that information at hand and essentially targeted markers that enable us to decipher that information, I think we'll see a lot of novel varieties emerge that are where we have targeted integration of particular traits, such as minor cannabinoid expression from, for example, wild or semi-wild population, and integrate that particular part of the genome into a commercial variety, so that we maintain all of the traits, such as all the agronomic traits of interest. So very exciting times. Here's just another side note where we're looking more at hemp varieties as opposed to drug-type cannabis. But what's interesting with this particular variety here, which is called X59, is also called the hemp nut. So a lot of the hemp seeds that we get in Canada is made from X59. And it's a certified variety, but what's interesting is that if you go into a field of X59, you'll notice several different phenotypes. In this case here, not only are these plants different sizes, so we've kind of characterized them as tall, medium, and short, but you notice a large difference in fragrance based on the size, based on the different phenotypes. So the shorter plants are much more fragrant and also exhibit, a lot of more medicinal or larger trichome production than what you see on the larger plants, and and essentially being able to take a variety like that and from seed be able to predict which phenotype it's going to fall into, possibly with a protocol that is non-destructive, which I don't know if it's possible for cannabis yet on the seed, because I think the seed coating is maternal. But it offers a lot of promises in terms of being able to have more consistent production, as well as more targeted production, depending on what the philosophy of the cultivator is. And then again, I mentioned variety verifications. The current offerings and a lot of the people that are doing this is, I think the last one I saw was 1.4 million SNPs from a Canadian company. Here I show that with 18 SNPs, you're actually able to tell varieties apart. So making all the either whole genome sequencing or genotyping by sequencing not the most parsimonious tool to use for these purposes. and again, one of the great things is this is something that can be done in-house at relatively low costs, and also I think very efficiently, given that you don't have to wait several weeks to get results back. You can get it in the same day. Thanks very much. Hope I didn't go too far, too long on my talk.
So, questions for Philippe?
So, when you say limonene, dominance, etc., what is in terms of percentage?
Okay, so when we're talking about a particular terpene being dominant in a variety, the number is typically 0.5%. But essentially, what you see, that's typically what it is. Very often, you'll see that some have more than 1% actually, but 0.5 is really the cutoff. You're welcome. And also, sorry, relative to all the other terpenes, it's the highest as well. Yeah, Manjot?
Looking at your results, we see cannabinoid biosynthesis genes also coming up in your model, which you tried for the terpene biosynthesis, right? So does it mean that cannabinoid biosynthesis pathway genes are also affecting the terpene biosynthesis pathway, and these models are to be built together rather than independently, considering all the genetic diversity available in cannabis?
Well, you probably know more about that than I do, but I can tell you that, you know, geranyl biphosphate, GPP, is a common precursor to both CBGA as well as a number of the terpenes. So, you know, there definitely is a relation there. The model is very simple and really just based on the terpene expression at this moment. Now with the map of the cannabis genome, if we're able to map these particular SNPs and how they interact with the final phenotype, I think we'll be able to find you know, some answers to that. But right now, it's up and coming stuff for sure. Yep.
So do you know what chromosomes your 23 Cas markers are on?
I have somebody that's mapping it as we speak, but I don't currently. I haven't mapped it to the CanSat 2 and CanSat 3 yet, but I will do soon.
Okay, thank you, Philippe.